So we're going to move into our last lecture on the adaptive immune response, and our goal with this lecture is really to understand how adaptive immune responses generate and get a big picture of how these responses go. And to understand adaptive immunity, we have to start off understanding the lymphatic system. Our capillaries are obviously high pressure containment systems, and fluid leaks out of those capillaries all the time. Plasma leaks out in particular. And this plasma, when it leaks into tissues, we call it interstitial fluid. And in order to maintain the balance of fluid within those tissues, we also have a system for moving fluid out of the tissue. And this system is called our lymphatic system. And so fluids can move into the lymphatic capillaries also via pressure and move along those lymphatic capillaries and back into the blood eventually. And the mechanisms of movement of that lymph fluid in the capillaries is first the movement of skeletal muscles, and second arterial pulsing kind of slowly pushes that fluid along the capillary. Backflushing is prevented because there's little valves all along the way of those capillaries. And the lymphatic system is important in a variety of ways. We know that it's important for delivery of large molecules and metabolites. It's important for maintaining fluid balance between tissues and blood. But really, for our purposes, we're studying immunology. The lymphatic system is incredibly important for the generation of an immune response, and that is because the lymphatic system is the way that antigens get from tissues into secondary lymphoid organs like the lymph node and the spleen, where all the lymphocytes are. Here's a picture of the lymphatic system within an adult. You can see that there are many lymphatic capillaries that lead into lymphatic vessels, that lead into lymphatic ducts. And all of these lymphatic highways really connect strings of lymph nodes. And a single capillary might collect a whole string of lymph nodes, one after the other. And these lymph nodes that are connected to certain tissues are often called draining lymph nodes because they are draining lymph that is collected from those lymph nodes. And lymph eventually migrates back into the blood via the thoracic duct, thus maintaining the balance overall. This is just a table that I've put up from your first aid book showing some of the important lymph nodes that are palpable during infection. Lymph nodes actually become enlarged during infection. This is called lymphadenopathy, and palpably large lymph nodes can be signs both of infection, can be signs of inflammation. In some cases it can even be signs of lymphoma. And so you might want to start familiarizing yourself with this table. It will not be testable for this material right now, but is useful information for you to begin to learn about. Now the lymph empties into the lymph node, and lymph that is entering a lymph node we call afferent lymph. Lymph that is exiting a lymph node we call efferent lymph. They're entering via the afferent lymphatic vesicle and exiting via the efferent lymphatic vesicle. Now, there's some really important structures here in the lymph node. One of those very important structures is the B-cell follicle, and I often refer to this as the B-cell zone. A second or a key feature of these follicles is you can have primary follicles that are dense structures comprising almost all naive B cells, but you can also have secondary follicles, and this is where B cells get activated and start to expand. And the place where this happens is the germinal center, and that is this area within the B cell follicle, usually light-colored area where B cells are undergoing expansion, and is surrounded by a darker area called the mantle zone, also rich in B cells. And then connected to these, or near these B-cell follicles, are the medullary cords, and these cords are actually rich in plasma cells, so much of the antibody production occurs within the medullary cords. Now there's also an area called the paracortex, shown here in blue, and this is where all the T cells live. I often call this the T cell zone. Also lots of dendritic cells in this area. There's a few other structures that you should know about that you should just kind of have a passing familiarity with. One of those is that the lymph node has a capsule The capsule also gives rise to these structures called trabecula. The lymph node is also full of or fully supplied by the blood via veins and arteries. And so the blood supply can come in, and this is how T cells actually enter into the lymph node via the blood. And then you have these medullary regions where medullary sinuses that are full of things like macrophages. Now, these are all important structures, but really when we talk about secondary lymphoid organs and lymph nodes, we're really going to focus on key structures like B cell follicles, the paracortex, and the germinal center. And these are the three that you really need to know about. Here's a picture of a lymph node, and you can see here that if you look down below, there is a primary follicle, densely packed naive B cells. Next to it, you have a secondary follicle. And if you look in that secondary follicle, you can see that inside that secondary follicle is a light-colored germinal center surrounded by a darker mental zone. You can also see these areas around the follicles. This is the paracortex, and these areas are rich in T cells. Now, the spleen is also a secondary lymphoid organ, and there are many key functions of the spleen. The spleen also has a capsule, also has trabecula, also has sinuses, but the spleen is basically composed of red pulp and white pulp. And while you will get more information on the spleen and spleen anatomy in other classes, for the purposes of the immune system, I really just want to focus on what happens within the white pulp. And so as you look at the spleen, you'll notice that it also has structures that look a lot like the germinal centers and the follicles in the lymph node. And so in this way, the spleen is highly analogous as a secondary lymphoid organ to the lymph node. And these follicles comprise the white pulp. And so here I'm going to label a follicle. And then next to it are these areas of pink surrounding the arterioles. And this is called the periarterial lymphoid sheath. And for short, it is often referred to as the PALS or PALS. And this is the T cell zone. And together, the follicle and the periarterial lymphoid sheath comprise the white pulp of the spleen. And these follicles have germinal centers. They have a mantle zone. They also have this marginal zone around the mantle zone. And this is a zone that's actually rich in a very specialized population of B cells, as well as a specialized population of macrophages. For future reference, we are going to refer to spleen and lymph node as secondary lymphoid organs, and we're not going to really distinguish much because the mechanisms between how adaptive immune responses are generated in each organ are highly similar. And so we're going to kind of describe immune responses in these two organs together. This is just a histological view of the spleen, and here you can see the PALS, and it's surrounding this central arteriole. And if you look below, you can see, and I should also mention outside the PALS, this is all red pulp. And you can also see two arterioles here in the other picture, and they're surrounded by these germinal centers, these B cell follicles. And there's an area of red pulp also that's visible in this one. So how do antigens get to secondary lymphoid organs? Well, we mentioned that spleens really drain blood antigens, but tissues are drained by the lymphatic system. And pathogens like bacteria and viruses can actually enter into the lymph directly and migrate directly to lymph nodes. And that also includes pathogen products like secreted proteins that can enter into the lymph and get into the lymph nodes. Antigens also have another way of getting into the lymph node, and that is that they can be transported there by cells of the innate immune system. And the cell type that does this is the dendritic cell. Dendritic cells are phagocytes, and they are constantly taking up pathogens. And when they are exposed to pathogens or to cytokines produced by things like macrophages, dendritic cells become activated. And instead of residing in the tissue, they change their location. They begin to migrate towards the lymphatic system, and they will actually migrate into the lymphatic capillaries and slowly migrate towards the lymph node, where they can present those antigens to T cells. And again, these dendritic cells are expressing MHC molecules and specialize in presenting antigens to T cells and activating them. So let's get a big overall picture then of how T cells and B cells are getting activated in the T cell zone and in the B cell zone. So lymph can drain into the T cell zone or dendritic cells. These cells can activate T cells, and when they activate T cells, these T cells undergo clonal expansion. T cells within the T cell zone that are activated by antigen can differentiate into cytotoxic T lymphocytes, or they can differentiate into T helper cells. And once they do that, many of these T cells actually will now migrate out of the lymph node into the blood, and they will migrate to tissue sites of infection. And there they will mediate their effector functions that we talked about previously. Now there's a subset of these cytotoxic T lymphocytes and T helper cells that don't migrate out, and in fact they become memory T cells. And lastly, there's a subset of T helper cells that instead of migrating to tissue sites of infection, actually migrates to the B cell zone of the lymph node. Now let's transit over to the B cell zone. In the B cell zone, you are also having a delivery of lymph, which leads to the activation of some B cells. And so B cells are activated by soluble antigens. Let's draw a picture of a B cell with antibodies on its surface, and these antibodies are binding to this virus that has been transported via the lymph into the lymph node. When this B cell gets activated, it becomes an antibody-producing cell and starts to undergo expansion, but most of the antibodies that it produces are IgM, and in fact can also differentiate into a plasma cell that can produce fairly decent amounts of IgM. But for a B cell to reach its peak function, it needs to interact with a T cell. So let's draw this B cell when we have a T helper cell that is now migrating towards this B cell within the germinal center. And this T helper cell will induce the enhanced activation of this B cell via activation signals and via the production of cytokines. And when this happens, it dramatically enhances the activation and the clonal expansion of these B cells. It leads to the formation of the germinal center, and it leads to the process of isotype or class switching, such that these B cells can now produce other types of antibodies, perhaps IgG or IgA or IgE. These B cells will also undergo affinity maturation via mutation of their antigen receptor locus, and thus be able to recognize their antigens with much higher affinity. What this all leads to is the development of plasma cells that can secrete IgG antibodies or IgA antibodies, and these antibodies are generally high affinity antibodies and much more effective in their anti-pathogen effector functions. Also important to note, memory B cells can result from this entire process. So that is an overview of how B and T cells get activated the first time. And this is a look here in this graph on the left of what happens during secondary and tertiary antibody responses. And you might have gotten a vaccine at some point where you had to go back and get boosted a month later or 2 months later. And these boosting events are meant to induce secondary and tertiary antibody responses. Whereas primary antibody responses are mostly IgM, secondary antibody responses are mostly IgE, and successive boosts of the antibody response leads to high titers or concentration of antibody and higher affinity antibodies. And this is really the basis for why we do vaccine boosting, and is the basis for protection by antibodies. On a cellular level, B and T cells, when they're responding the first time to antigen, undergo a period of clonal expansion. And after a week or two, they reach their peak clonal expansion. And once the pathogen is cleared, most of these B and T cells die off, but a small proportion of these B and T cells give rise to long-lived memory B and T cells. And these memory B and T cells, when they see the pathogen a second time, respond faster and more robustly. And this is the cellular basis for the memory response. Whereas a primary response by the adaptive immune system is delayed, a secondary response by the adaptive immune system is immediate. So key study points from this lecture: you should review the function of the lymphatic system and the mechanisms by which it delivers antigen to lymph nodes. Also review the anatomy of secondary lymphoid organs, particularly lymph nodes and spleen, and understand the mechanisms that lead to the induction of an adaptive immune response in the lymph node. And lastly, review the features of a secondary immune response and how that differs from the primary immune response.